Hello, my name is Kimberly Puente, and today I'm going to be talking about mitosis. So, what is mitosis? Mitosis is a type of cell division in which one cell, known as a daughter cell, divides to produce two new cells, which are known as the daughter cells, that are genetically identical to itself. In the context of the cell cycle, mitosis is a part of the division process in which the DNA of the cell's nucleus is split into two equal sets of chromosomes. Most of the cell divisions that happen in our body involve mitosis. During development and growth, mitosis populates an organism's body with cells, and throughout an organism's life, it replaces old, worn-out cells into new ones. The main goal of mitosis is to make sure that each daughter cell gets a perfect full set of chromosomes. Cells with too few or too many chromosomes may cause the cell not to function well. They may not survive, or it may even cause cancer. Because of this, when cells undergo mitosis, they don't just divide their DNA randomly and toss it into piles for the two daughter cells. Instead, they split up their duplicated chromosomes in a series of steps. Mitosis consists of four basic phases: prophase, metaphase, anaphase, and telophase. These phases occur in a sequential order, including cytokinesis, the process of dividing the cell contents to make two new cells, and it starts in anaphase and telophase. During interphase, which is this one right here, the nucleolus and the nuclear envelope are distinct and the chromosomes are in the form of thread-like chromatin. This would be the chromatin, and then the nucleolus would be this purple circle, and the nuclear envelopes, which are these. In early prophase, which is this one right over here, the cell starts to break down some structures and build others up, setting the stage for division of the chromosomes. The chromosomes start to condense, making them easier to pull apart later on. The mitotic spindle begins to form, and the nucleolus, or nuclei, which are a part of the nucleus where ribosomes are made, disappears. This would be a sign that the nucleus is getting ready to break down. In late prophase, which occurs here as well, sometimes also called prometaphase, the mitotic spindle begins to capture and organize the chromosomes. The chromosomes finish condensing so that they are compact. The nuclear envelope breaks down, releasing the chromosomes, and the mitotic spindle grows more, and some of the microtubules start to capture chromosomes. Next, we have metaphase, which is this one right here. The spindle has captured all the chromosomes in this phase and lined them up at the middle of the cell, ready to divide, which is this line right here. At this stage, the two kinetochores of each chromosome should be attached to microtubules from opposite spindle poles. Before proceeding to anaphase, which is this one over here, the cell will check to make sure that all the chromosomes are at the metaphase plate with their kinetochores correctly attached to microtubules. This is called the spindle checkpoint and helps ensure that the sister chromatids will split evenly between the two daughter cells when they separate in the next step. If a chromosome is not properly aligned or attached, the cell will halt division until the problem is fixed. In anaphase, the sister chromatids separate from each other and are pulled towards opposite ends of the cell. So here they show that they are separated versus over here they are all together. The protein that holds the sister chromatids together is broken down, allowing them to separate. Each is now its own chromosomes. The chromosomes of each pair are pulled towards opposite ends of the cell. Microtubules not attached to chromosomes elongate and push apart, separating the poles, making the cell longer. In telophase, which would be this one right here, the cell is nearly done dividing and it starts to reestablish its normal structure as cytokinesis takes place. The mitotic spindle is broken down into its building blocks and the new nuclei form, which are these right here. One for each set of chromosomes, nuclear membranes and nucleoli reappear. The chromosomes begin to decondense and return to their stringy form. After telophase, cytokinesis, which is the division of the cytoplasm to form two new cells, overlaps with the final stages of mitosis. It may start in either anaphase or telophase, depending on the cell, and finishes shortly after telophase. When cytokinesis finishes, we end up with two new cells, as shown here, each with a complete set of chromosomes identical to those of the mother cell. The daughter cells can now begin their own cellular lives and may undergo mitosis themselves, repeating the cycle all over again.
Okay, so I am going to go through the phases of meiosis. So meiosis is a lot like mitosis. It starts with interphase, but remember, the point of mitosis is that it makes two individual cells. And in meiosis, what we're trying to do is match 4 genetically different cells because they're designed to be sperm and egg. And so that's basically the whole point of sex. So let's begin at the beginning with interphase. So this is just, so as the cell is it's being formed, and as you can see right here in the nuclei, we have those 2 pair of chromosomes. And if we look at the centriomes, we just have one centriome right here, one centriome. And so that's usually what a cell looks like. And what is going to happen here is you can see in the interphase, we've duplicated these centriomes so that we have 2 of those. And the DNA is also all loose. So it's not really tightly wound up into these chromosomes, as is usually what you see. So they usually look like X's, as so, which means that during the S phase of interphase, we've copied the DNA. So we have 2 complete sets of DNA. And this looks identical to mitosis, but it's just about to change. Okay, and so what happens next is prophase, right here. During prophase 1, the chromosomes undergo what's called synapses. And so what's happening is chromosome 1, you get chromosome 1 from dad, and then you get chromosome 1 from mom. And they both come together, and so they're both wrapping around each other and really tightly. And what's really going on is that they're swapping parts of their chromosomes. In other words, these are pretty much identical except for the crossing over. So segments of the chromosome from mom are switching with chromosomes from dad. You can see right there. Same thing over here, and the same thing. So that's given us a variation. If this didn't occur, synapses didn't occur. The chromosomes that you get from your mom and your dad, you would give to your children, either as chromosomes from your dad or vice versa. And what we're doing is crossing over, is we're combining the 2 chromosomes that we get from our 2 parents and making a brand new chromosome that we want to deliver to our child. That's the importance of this. If we keep on going, then we're going to move to metaphase 1. What's happening in metaphase 1? So you can see that they are all lining up or meeting. So that's what's the most important thing that's happening in metaphase 1 is that they're lining up, and that is called a metaphase plate. And so we have 4 different ways that the chromosomes could organize themselves in a differently at metaphase 1 right here. And that is giving us a variation of 2 ways that the number of pairs possibility. So how could they arrange themselves independently? Orientation of metaphase 1. In humans, we have 23 chromosomes, and so there are over 8 million ways that all of those chromosomes could independently ordinate themselves during metaphase 1. And so that's what we're getting again, and that crossing over is giving us a huge amount of variation in meiosis. And one sperm has to find that one egg. And so it's really over around 60 trillion possibilities of an offspring just based on independent orientation itself. So another thing that happens in metaphase 1 is that spindling is going to attach. So you can see that in the centriomes move to each of their sides of the cell. And the spindling attaches to the centriomes of each of those. Yes. So, okay. And during anaphase 1, it is pulling them apart. So that's also what anaphase is most known for, is for the pulling of the apart. So we can see that those chromosomes are going to either cells. And then during telophase 1, what's happening is we're reforming a new nuclei at each side. So we're dividing the nucleus, which is meiosis. But now we have to divide the rest of the cell, and that's called cytokinesis. and right there, cytokinesis, it says right there. And so we're done with meiosis 1. We've gone through the prophase where we saw the synapse. We went through the metaphase where we had the independent orientation, and now we've divided the nuclei into each of those cells. So what's going to happen next? So they basically look identical to, it basically looks identical to mitosis, but the difference is just about to change. And what happens next is we're going to go through prophase 2, right here, prophase 2. Okay. During prophase 2, there's no more crossing over, but what's going to happen in those 2 chromosomes are going to line up again. They're going to meet right in the middle and spindle. If it's going to attach to each other of those centromes now, if we look at what happens, each of those centromes are going to be pulled to each other's side. So they're going to be pulled to each other's side, and during anaphase 2. And then finally during telophase 2 and cytokinesis 2, we've created those 4 cells that we wanted in the beginning. And so if we look at where we began, we had 4 chromosomes, or 2 pairs of chromosomes. Now we have 4 cells, and each of those cells only have 2 chromosomes. And so what would happen next in the cycle of life is, if these were sperm, they would fertilize in an egg, and then we would start over again, and we'd have a brand new organism. That's what's going to be created through mitosis. Now, this is how we make sperm. Each of the sperm are going to be made like this. Again, there would be a way more chromosotic because we have 20 chromosomic in us, because we have 23. It's a little different with the eggs because there is so much importance in the cell that only one of these nuclei will actually be used, and the other ones won't be used generally in the cell. And that allows us to keep all the important parts of the cell